Hi, I'm Dan Knights. Welcome back to Discovering Patterns in the Microbiome. Today we're going to be talking about assigning taxonomy to marker gene sequences. And this is one of the first steps in processing data. It's typically done after the sequences have been demultiplexed and separated into their different original samples. And often after we've done OTU picking as well, although it doesn't necessarily need to be done that way. We're going to be assuming that we're using the 16S rRNA gene and that we have some variable region that was amplified out of that gene. I've talked about using primers to amplify out a piece of this gene in the past. Here are a couple examples of different primers. So these were primers that were used in the Human Microbiome project. There were actually three sets of primers used in this project. The first set flanked the V1, the first variable region, and the third variable region. So it actually spans V1 through V3. The next set spanned V3 through V5, and the next set spanned V6 through V9. And so here you can see when you actually read about a primer, they're named by the index into the 16S gene at which they start. And that's using usually this reference 16S that's from E. coli. So you can see that 27F534R starts near the beginning and goes about a third of the way into the 16S. The V3, V5 region starts before the end of the V1, V3, because it's starting before the V3 variable region, and goes out to about the 926th residue. You can actually get all of the V1, V3, and V3, V5 data from the Human Microbiome project. It's available at hmpdac.org. It stands for Data Analysis Coordination Center. And I wanted to show an example from these data. It was a unique opportunity where lots of samples had been run through two different sets of primers. And what this plot is showing is if you count up how many OTUs were recovered within a particular species or a particular genus using V1, V3 or using V3, V5, there are some pretty big differences. So for example, down at the bottom here, Coprococcus looks like it had about 64 different OTUs recovered from the V3, V5 data, with only 2 or 3 recovered using the V1, V3 data. So already this is really bad news because choosing which variable regions you're going to amplify has a huge bias on what's going to come out biologically at the end of all of the data processing. So this should make you very concerned about combining data between data sets where there have been two different sequencing methods used. One of the most popular primer sets in use right now is 515F806R. This is used in the Earth Microbiome project. The protocol is very conveniently put up online, and the nice thing here is that, so this spans the fourth variable region. Fourth variable region tends to give decent coverage of most of the bacteria, although some get under-amplified. And the nice thing is that when you use a MySeq run with 2x300 or 2x250 length reads, they will actually overlap in the middle. Because the two primers are close enough together, then you can stitch them together and get actually a little bit longer read. And at the point at which they stitch, you can resolve some of the ambiguities. Now, today we're talking about what you do when you've amplified out one of these regions and we want to assign taxonomy to it. And you could think of this as a standard classification problem from machine learning where you have a whole bunch of training data. You want to learn a model that maps some input data to a label, and then you're going to run that model on a bunch of new data. So here the training data are these DNA sequences, And the labels are the taxonomic labels, where we know sometimes all the way down to the species level, sometimes we only know down to genus level, and so on. Then we learn a model that is a function that maps from the input sequences to the taxonomy. Then we run that on the new data, which would be this query sequence. And then the output should just be a taxonomy assignment and perhaps a confidence level assigned to that taxonomy. There are many different ways of doing this. Here's a little diagram from a review a few years ago. There are even some more ways that have been published since then. The general classification is that you can either do something based on alignment, so you're trying to align every input read or every input sequence to a reference database, or there are a number of techniques that use k-mers. So k-mers are just very short sets of nucleotides, say 8 nucleotides long, and we're going to talk more about how those are used for the classification later. Here's a comparison from that same paper of using some of those different methods in a number of different data sets, and what the taxonomic labels looked like. So in this picture, each box is a different set of, it's a different data set. So the box on the far left is from the Guerrero-Negro data set. This is a microbial mat that's very thick and very diverse. The middle box is from the human gut, the Human Microbiome Project, and then, actually some different samples. And then the box on the far right is from mouse gut. So I want you to take a moment, and then every row within each box is a different taxonomic classification method. So I want you to take a moment and think about which, if you look, say, just at Guerrero-Negro, and you look at the different rows in there, what conclusions can you make about taxonomic classifiers? So I invite you to pause the video and think about what that figure tells you about different taxonomic classifiers. So one of the conclusions that you might have made is that there's a lot of variability between different classifiers in how they assign taxonomy. So in the first one, in the first three, you can see that the samples are relatively consistent in which bugs are present in the samples, meaning that you see kind of a straight line going across for each different bug. Whereas the last three classifiers have a lot of variability. So for example, you'll see those purple blobs mean that there's a particular species or a particular genus that showed up in one sample, then not in the next one, then it showed up in another, and so on. And that particular genus doesn't show up in, or sorry, this is phylum, doesn't show up in any of the other rows. So there's a lot of variability between methods in terms of which species are called. And then you see that in human gut and mouse gut as well. You might also notice that some of the classifiers, those bottom three, across different environments tend to recognize a lot more species than others. So the method that you choose to use for taxonomic classification is very important. Now, I'm going to talk about the most common method, which is the second row here, which is the RDP classifier. So again, the RDP classifier is treating this just like a machine learning problem where we have training data and labels. And it's using what's called a naive Bayes classifier. This was published in 2007 and still remains the most commonly used approach for assigning taxonomy. To talk about this, we're going to do just a little bit of probability basics. I have a few terms up here. The prior probability just means your belief that a certain thing happened before you have any new information. We have conditional probability. So that's the second row. P, we would say P of x1 given x2. The way you'd read that is the probability of x1 happening given that you know x2 happened. And then joint probability, so that's P of x1 comma x2, that means the probability of x1 and x2 happening at the same time. So these x's just stand for events, like it rained or I won the lottery. And we're going to briefly discuss Bayes' rule, which is what's used to do the classification in the RDP classifier. So Bayes' rule has turned out to be extremely useful in machine learning because it kind of flips the learning problem around in a way that makes it more tractable. And the basic setup is this. If you have you want to know the probability of a certain statement being true, like for example, a certain taxonomic label, given all of your training data and whatever input data there was, you can flip that, so that's a conditional statement. You can flip the conditional statement around and say that the probability is equal to the probability of seeing the data, assuming that the taxonomic label is true. So that's the P of x given C, times the probability that this is just the background or prior probability that that classification or taxonomic label is true, all divided by the probability of seeing that data. So this is just a mathematical equivalence. And if you want to talk about this to a statistician or a machine learning person, you would call the left side of the equation the posterior probability. Then the right side, the first term is called the likelihood. So that's the likelihood of the data given the label. The next one is the prior. So that's the prior belief in the label before you've seen any of the new data. And then on the bottom is what's called evidence. Bayes' rule, if you ever forget Bayes' rule, this is a little trick that I use to remember it. You've all seen Venn diagrams before. So what we have here is a circle showing all of the probability of this event called A. That's the red circle. The blue circle is all the probability of the event called B. And there's some overlap in the middle, which is the intersection when both A and B happen. So that could be it rains today and I brought my umbrella. We could all agree, I'm sure, that if I want to specify the probability of A and B happening together, so that's that little slice in the middle, you could rephrase that as the probability of ending up in the B circle somewhere, that's the P of B here, times the probability of being in the A part of that greater B circle. So that's the P of A given B. And equivalently, we could write it as P of B given A times the probability of ending up in the A circle. And if you take the right two parts of this equation and divide by the probability of B, divide by the probability of B here, you end up with Bayes' rule. Let's make this a little bit more concrete. So here's how Bayes' theorem is used to classify taxonomy. And I think it's worth looking at this because there's probably quite a bit of room for improvement here. So the way that we're doing this is we're trying to assign what genus a sequence is from, given that sequence. We use Bayes' rule to rewrite this as the probability of that sequence given that you're in the genus. And that we can figure out from our training data. How likely is this particular DNA sequence to show up in a particular genus? So that's the likelihood. So that's easy. The prior probability is just how likely is that genus in your data. And on the total probability, or the evidence down here, is the probability of seeing that sequence overall. The neat trick that is used in machine learning is that you can actually ignore some of those terms. So let's look at an example. Let's assume we want to know what's the probability of a particular genus we'll call G, given this sequence TTCAG. Now, the way that the RDP classifier works is it breaks the sequences down into what we call k-mers. So these are just little sections of DNA. And for demonstration, we're going to call k equals 2. But k could be 8, for example. 2 would usually be too small. So the way the probability works out is that the probability of the genus, of being in a particular genus, if you see that sequence, is the same as the probability of seeing the first, we're going to pretend it's the same as the probability of seeing the first 2 letters, if you're in the genus, times the probability of seeing the next 2 letters, if you're in the genus, times the next 2, times the next 2. So those are all conditional probabilities. And then at the end, we have the background probability of the genus, and at the bottom, we have the background probability or prior probability of that particular k-mer. So the trick that's used is that we don't really care about the probability of that particular sequence, so that's the bottom of the fraction, because we only care about the relative probabilities of all of the different genera for that sequence. So the denominator is going to be the same when we're assessing all of the different genera for that sequence. So we can cross off the denominator. The other trick that people use is to just forget about the prior probability of the genus, and we'll assume that there's a uniform probability of seeing different genera in an environment. So that's one place where there could be a major room for improvement, because you're much less likely to see like some Bradyrhizobium genus in a human gut. And if we put a prior probability distribution on each of the species, knowing how common they are in human gut microbiomes, we could probably dramatically decrease false positives in taxonomic classification. So ignoring this term here is probably not the best way to do taxonomic classification. However, that's an open research project. So the way we would do this in practice is we would just we would cross out those 2 parts of the equation. We'd look in our training data and see how common TT is in this particular genus. Let's say it happens 10% of the time, and so on. And we multiply them together, and we get a posterior probability. Then we do the same thing for another genus, say G2, and we get a posterior probability. And then we just see which one is larger. And whichever one is larger wins. So that's the classifier. Here's the estimated performance of this classifier. This is from the original publication. And this is showing how often it classifies, it identifies the correct taxonomy, depending on the sequence length and depending on what taxonomic level you care about. So what can you see in this figure? Well, if you look at the very back of the figure, that's showing that if you have really long sequences like 400, or actually in that case it's full-length 16S sequences, and you only care about classifying at the phylum level, you can get 99.8% of the classifications correct. However, if you have short reads, so short reads might be, say, 200 bases long, and you care about classifying down to the genus level, we're only getting 83.2% of the sequences right. And I think we can do better than that. One nice thing about the RDP classifier is that when it gives you a taxonomic assignment, it also gives you a confidence level about that taxonomic assignment. So the way it does this is it just chooses a random subset of the k-mers in a particular genus, and it repeats the training and it repeats the classification, and then you just count up how often you see the same taxon with those random subsets, and then the confidence is reported as a fraction, like 0.8. So typically, what people do is they'll run RDP, and then they'll take all of the classifications that were at 80% confidence or above. Now, as I mentioned, I think this is an area for a good research project. If you think of a typical supervised classification task, this is no different, except here we have 5,000 classes or more, so those are all the different species that you might typically have in a reference database. You have sometimes 5, 10, 20 examples in each class, but you also have a very carefully defined hierarchy relating the training samples to each other. That's the taxonomic hierarchy. The state-of-the-art solution is still more or less naive Bayes classification, but that could be improved upon, as I mentioned. So in summary, classification is typically done using the RDP classifier and naive Bayes. I think we can do better than that. Actually, there was a more recent publication showing that the random forest classifier seems to work better than RDP or than naive Bayes, which is not surprising, although I haven't seen this in use much yet. In my opinion, the reference databases are always getting more comprehensive. So eventually we'll get to the point where pretty much everything in our samples is represented in the reference database. And this is especially true for human gut microbiome, for example. And in that case, we probably don't need to learn some complicated function that maps k-mers or other smaller features that were extracted from the sequences. Instead, we can just take the full reference sequence, the full input sequence, align it to every sequence in the reference database, and find the best alignment. So that would be called a nearest neighbor approach, but it's going to be important to have optimal alignment there. And there's another major issue, which is that when you're using just one variable region, say V4, you might have a whole bunch of species that have almost exactly the same sequence there. So what you really want to do if you're using this nearest neighbor approach is probably to find every single hit that's above, say, 99% in your reference database, and then look at all of their taxonomies and find out how far down they agree. So if you get, say, 5 different hits from the genus Bacteroides in 5 different species, you probably don't want to call any one of those species. You probably just want to call the genus Bacteroides. And that would help you control for false positives. So I think moving forward, using optimal alignment and full reference databases is going to be the way to go. And just want to close with one other thought about what other information we can use than the actual DNA sequence when we're trying to do the assignment of taxonomy. So if you like, I invite you to pause for a second and think about, is there any other information we can get out of the input sequences and out of the training data sequences that would help us do the taxonomic assignment? There might be other answers, but the one that I was thinking of is the secondary structure. So there hasn't been a lot of use of secondary structure. So that means, if you recall, the ribosomal RNA, when it gets transcribed off of the genome, off of the DNA, it folds up on itself into those complex secondary structures. And it would be interesting to find out how well the secondary structure, using secondary structure can improve the taxonomic classification. There has been a little bit of work done in this area, but I haven't seen it been adopted into common usage.